friends, in this video we will discuss about nuclear protein targeting. We all of us know that protein are synthesized inside the cytoplasm, and some proteins are also need inside the nucleus. We all of us know that, like histone proteins are need inside the nucleus. Then how these proteins synthesize inside the cytoplasm and it will target inside the nucleus. Okay. Then transcription is also occur inside the nucleus, and then mRNA comes inside the in the cytoplasm, and these mRNA attached with the free ribosomes of cytoplasmic ribosomes, okay, and translate to form protein, and these protein go inside the nucleus again, okay. Then these proteins target inside the nucleus by the help of some signal sequences and translocan at the nuclear envelope, okay. But here translocans are very specific in the nuclear pores, okay. These these translocans are very specific. Now we discuss. Let us draw first. This one is outer membrane and this one is inner membrane of nuclear envelope, okay, nuclear envelope, and this one is perinuclear space, okay. This one is perinuclear space. Here some protein, basket like protein present, okay, and this one is cytoplasmic filament, okay, and here this one is central channel, cytoplasmic rings are also present here. This one is nuclear ring, okay, and this is cytoplasmic ring. This one is cytoplasmic ring, okay. Here some another protein is also present. And this will attach with the specific protein. These are the some specific protein. Okay. This is spoke complex. This one is spoke complex, and this one is luminal ring. Same is here. Okay. And this is the nuclear basket. This is known as nuclear basket. Nuclear basket. Tick. One of the specific protein also present. One of the specific protein also present at the inner membrane of the nuclear envelope, which is known as lamine protein, which provide toughness. Okay, it provide, this protein is very specific, which provide toughness of nuclear envelope. Okay. Now, how proteins are target inside the nucleus. This one is nucleoplasm, and this one is cytoplasm. We all of us understood cytoplasm. Okay. This one is nucleoplasm. Okay. Now we discuss about, this is the normal structure of nuclear pore. Okay. Now we will discuss about protein targeting yet or transportation inside the nucleoplasm. Okay. We consider these are the nuclear pores. These are the nuclear pore. Both are nuclear pore, like this. These nuclear proteins are present, complexity. Actually, these proteins are known as nuclear protein, which are about 300 to 400 proteins. Different types of protein makes a complex together at the nuclear pore region. Okay, to form nuclear pore. Now we will discuss about protein transportation inside the nucleus. Okay, protein transportation. There is two types of protein, that is exporting and importing. Exporting means that protein which wants to go to the cytoplasm, and importing means import inside the nucleoplasm. Okay, let us consider this one is exporting protein. Okay, and this one is importing protein. Okay, now in exporting protein interact with the NES protein. Okay, NES protein, nuclear exporting signal molecule. Both are interact together and go interact together and go to the outside of nucleoplasm. Okay, here exporting protein freer. Exporting protein will free to the NLS, and NLS is recycled back. NES is recycled back inside the nucleoplasm. And now importing protein is interact with the NLS, nuclear localizing signal molecule, and this complex molecule comes inside the nucleoplasm, and here exporting protein will free. Okay, now we know that these are the active transport protein, and active transport protein guide with a specific type of protein which is known as RAN. The concentration of RAN protein with GTP is high inside the nucleoplasm, and it help in exportation, with the hydrolysis of GTP. Okay, hydrolysis of GTP, and one of the another protein help in hydrolysis of GTP, that is GAP, GTPase activating protein. This one is GTPase activating protein. Now the RAN, here the concentration of RAN GDP is increases. Okay, now this GDP again recycle back inside the nucleoplasm, and again GDP is converted into GTP with the help of GEF protein, that is guanine nucleotide exchanging factor, which exchanges the GDP to GTP. Okay, it is all about the nuclear transportation. Okay, actually proteins are synthesized inside the cytoplasm, and again it will go inside the nucleus. We all of us know it. Okay, but these factors are responsible for the exportation and importation in the nucleoplasm. This is all about nuclear protein targeting, and this is small I want to show about the nucleus complexity. This protein is important for the nuclear pore structure, which is known as laminin lamine protein. Okay, thank you friends.